Okay, this is your last set of notes for the week of vacation. So we've talked about genetic control in prokaryotes, talking about it in eukaryotes. So again, we've seen this slide, right? Important differences are eukaryotic cells have a ton more genes. There's more physical obstacles in looking at genetics because the DNA is wrapped around histones and wrapped into chromosomes. And there are more non-histone proteins that are used in eukaryotic gene expression. So there's just more proteins, more DNA in general. Almost all cells in an organism are genetically identical, or what we call totipotent. Essentially, almost all of my cells have the same DNA. Obviously, sperm and eggs would vary, but everything has my same DNA. The differences between cell types results from differential gene expression, the expression of different genes by cells with the same genome. So I have the same, all the DNA is the same, but the genes that are expressed will determine what the identity of that cell is. Errors in gene expression can lead to diseases including cancer, and gene expression is regulated by a crap ton of things. And so we're going to go down the list of some of the things that regulate, out of all of my DNA, what is expressed and what is hidden in each cell. So again, remember that my DNA is, during metaphase and during division, basically created or wound into chromosomes. We go from our double helix around histones to make nucleosomes, into chromatin. We make basically looped domains into supercoils, and we actually get our chromosome. So there are certain lengths of DNA and RNA that are repetitive. They can be tandemly repetitive, repeated units at a site that are identical. Think about like the ends of DNA. They're not coding for anything, they're just repetitive. So out of like our mammal DNA, 10 to 15% of all of our DNA is just repetitive portions. And so that can be basically just sequences of letters that don't code for anything, they just take up space. Interspersed repetitive DNA, it are copies that are very similar but not identical. They still code for the same stuff. That's about 25 to 40%. So on average, 50% of our DNA is non-coding DNA. It's just repetitive DNA that is there. They definitely have a purpose. They just don't know what that is yet. So we have learned how to alternate the genome or the expression of genes to actually regulate them. So for example, there's three types. Gene amplification. In amphibian eggs, there are over 1 million copies of RNA made from the tiny circles of DNA. Basically, the more DNA I make, the more I will see a certain gene expressed. Gene loss in an insect, we call them gall midgets, during development, all but 2 cells lose 32 of their 40 chromosomes during the first mitotic division. These cells that retain the 40 will produce gametes, and so we can determine what genes are expressed through that. We also see transposed genes. The hemoglobin gene, which is the gene that carries your oxygen, has been duplicated, mutated, and transposed to other chromosomes to produce multiple but different copies of the same gene. And so we see a lot of shifting of genes before birth even. Again, so this is looking at the very first hemoglobin gene or the globin gene versus all of the different varieties that we see now. Basically, the chunk of DNA that codes for hemoglobin has been mutated just a tiny bit and moved to different places, and it'll make all sorts of different things. Another example of this, B lymphocytes, so our immune system cells, can produce all different types of antibodies that react with millions of different antigens and diseases. This happens because we can rearrange our AB genes. And so every time the sperm and egg are made, these genes are rearranged a little bit, and so this is why some people are immune to certain diseases. So when we're looking at gene expression, we look at epigenetics, which refers to the processes that influence gene expression or function without changing the DNA sequence. So the past couple examples we've looked at have actually changed the DNA sequence, but there are other ways that we can determine, or we can either make a gene be expressed and therefore affect the phenotype, or keep it from being expressed, also impacting the phenotype. And the two types are acetylation and methylation. Acetylation of lysine, lysine is something found in the histones, decreases the affinity of histones for DNA and other histones, therefore making the DNA more accessible for transcription. Remember that histones are the proteins that DNA wind around as it's getting compact, and so if it doesn't wind up, it is accessible for transcription. So acetylation will make a gene more likely to be transcribed than it normally would, leading to the gene being transcribed, the protein being made, therefore impacting the phenotype. Methylation is going to be the addition of a methyl group, adding to cytosines that are followed by guanines. So we call them like a CG-rich region. Methylation of those CG regions in a gene represses the gene expression, so then we don't make the protein, therefore changing the phenotype. Basically, it creates this like block of CGs that have a methyl group added to it, and it blocks the transcription of it. So we also have transcription factors, and we know that transcription is going to be creating RNA, and there are certain factors then that can influence whether or not something is transcribed. So we'll skip over these images for now. Here we go. So this is what's important. This illustrates how transcription factors, we could also call them activators, work on a certain gene. Essentially what will happen is before a gene, there will be a sequence of DNA where an enzyme or a protein can bind. And lots of different things can happen. An activator protein can bind, making the gene become transcribed. An enhancer, which might make it be transcribed faster or slower. There are also sequences of DNA that allow for you to control that. Basically, if we look in the top left corner, I would say that transcription factors, they are also like control elements. So there are spots along the DNA where things can bind to either inhibit or accelerate transcription of something. For example, in lactose persistence, the LCT gene produces the enzyme lactase, which digests lactose, similar to the if we remember the lac operon in prokaryotes. Lactose is found in dairy, and the LCT gene needs a regulatory protein, and so basically we have a protein that can bind to one of the control elements, which will either allow the gene to create the lactase, which is going to be the enzyme that digests lactose, or not. Most humans, after weaning, cease to produce the regulatory protein by a mutation in the gene that will allows us to continue making lactase into adulthood. In the mutation, you could have lactose persistence. Basically, there was not a binding to the control element, which changes our ability to process lactose. So other ways that we can alternate gene expression, we can see after transcription, we can see alternative splicing. If we remember back to transcription and translation, we saw splicing out of the exons and the introns. Depending on what is spliced out, we will see different phenotypes, different genes made. Exons and introns, right? This stuff, if you have a question, pause and look at it, but I did talk about this earlier this week. Again, same idea, multiple times. Other ways that we can impact gene expression once the RNA is made. So everything up until the last one was stuff that impacts the DNA. What can happen is mRNA molecules don't remain functional forever. Right? So when I go through transcription, that mRNA strand isn't going to work for forever. The length of time can affect the number of proteins that it makes and how long it makes those proteins for. Essentially, what will end up happening is along the way that DNA will start to decompose, and either no more of the protein is made or a different protein is made if part of the RNA decomposes. And that can control what proteins and how much proteins are made, impacting our phenotype. These are all examples of that. Other factors that can affect the genes post-transcription, so after we made our RNA, chemical signals that regulate the mRNA leaving the nucleus. We know that we go transcription in the nucleus, translation outside, and depending on when it leaves or goes out there can impact when it's made or when the proteins are made. Degrading the mRNA, that affects its lifespan. And we talked about that earlier, that as the mRNA degrades, the proteins, the proteins made will be impacted. Other examples of that, ubiquitin is a enzyme that can impact or accelerate the speed of degradation of the mRNA strand, basically accelerating it. Ubiquitin proteins will come in, it will bring in a ligase that will attach, and it will help degrade it faster. Other factors that can affect it post-translation. So we've done in the DNA, post-transcription, post-translation. So once we have a protein made, we can modify that protein. Often amino acids can be removed from the two tail ends for the protein to be functional. Proteins are often modified, so we add on little groups, and those can impact the proteins as well. And that is going to be it. That was super fast. I know, ask lots of questions about this because it is very complicated. And you're done for the week.